Inside every single one of your cells, right now, there are literally billions of tiny biological machines. They're working nonstop, building, repairing, and basically powering your entire body with incredible precision. But what happens when one of these perfect little machines starts making mistakes?
Well, let's meet our main character, an enzyme called EcoRI. And you can just think of it as a pair of molecular scissors or maybe even a tiny scalpel. Its main job is to patrol our DNA and make very, very specific cuts. It's a critical tool, not just in nature, but in the science lab too. And when I say it's precise, I mean it's unbelievably precise. Under normal conditions, its error rate is basically 0. This perfection is exactly what makes it so useful, but it also sets us up for a really fascinating mystery. So to really get how things can go so wrong, we first have to understand what it's supposed to do when everything's going right. This enzyme has one job and one job only. It scans through what could be miles of DNA looking for this one exact 6-letter sequence, GAATTC. When it finds it, and only when it finds it, it makes the cut. If the sequence is even one letter off, it just keeps on moving.
Okay, so let's get into it because this supposedly perfect machine, it has a secret. It turns out that under the right, or maybe I should say the wrong conditions, it starts to get, well, sloppy. See, scientists started noticing that if you just tweaked the chemical environment around the enzyme a little bit, something weird would happen. So they asked a simple question. What if we just change the soup that it's swimming in? And here is our mystery. Normally, EcoRI is a total specialist, right? Only GAATC. But when you add these things called osmolytes, molecules like glycerol that mess with the water around it, all of a sudden, it just lowers its standards. It starts cutting sequences that are kind of close, but not the right one. This rote behavior, that's the mystery we need to solve. This whole phenomenon actually has a name. They call it star activity. And the original research gives us our first huge clue. It's attributed to the, quote, dehydration of the protein-DNA interface. Now that sounds pretty technical, but all it really means is that it has something to do with water being taken away from where the enzyme and the DNA meet. But how do you watch something like that? I mean, this is all happening at the scale of individual atoms. You can't just use a regular microscope for this. You need a virtual one.
And this is where the really high-tech detective work begins. Scientists use these incredibly powerful computer simulations to build a perfect digital replica of the whole scene. The enzyme, the DNA, all the water around it. And this let them watch, frame by frame, exactly how every single atom was moving and interacting. It's like a crime scene investigation at the atomic level. So let's follow the clues and see how this all connects. Because what these simulations found wasn't just a single culprit. Nope. It was a chain reaction, a kind of molecular domino effect that ends up tricking the enzyme.
Here's the very first domino to fall. Remember those osmolytes we added? Those glycerol molecules? Well, they act like tiny little sponges. They get in there and they push the water molecules away from the surface, where the enzyme and the DNA are trying to meet. They're basically drying the place out.
And here's what's so cool about this. The simulations are so precise, you can literally count the missing water molecules. With just a 5% concentration of glycerol, about 379 water molecules get pushed out of the way. You double that to 10%, and that number almost doubles too, to nearly 700. This is our clue number 1. The crime scene is being dehydrated. Okay, so the water is gone. What happens next? Well, the DNA itself starts to change its shape. And this is the second absolutely critical domino to fall.
Now, this graph might look a little complicated, but the story it's telling is actually pretty simple. The red line shows the shape of the correct piece of DNA when the enzyme grabs it. See how it has that special kink in it? That kink is the signal. Now look at the other lines. When you add the osmolytes and take away the water, the incorrect DNA starts to bend and contort into almost the exact same kinked shape. The lack of water is basically forcing the wrong key to bend into the shape of the right one. And that brings us to the final piece of the puzzle. That fake kink in the DNA, that's all the enzyme needs to get fooled.
And this right here, this is the smoking gun. On the left, with plenty of water, the enzyme just can't get a good grip on the wrong DNA. But look at the images on the right. After that DNA gets kinked, all these new chemical bonds, think of them like tiny little handshakes called hydrogen bonds, can suddenly form. The enzyme feels those handshakes, thinks, aha, this is the right spot, and gets ready to make its cut.
It's been completely tricked. So now we can put it all together. We followed all the clues and we can lay out the entire molecular con job from start to finish. So here it is, the whole 4-step process. First, you add the osmolytes and they suck the water away. Second, that dehydration forces the wrong DNA to bend. Third, that new bent shape lets the enzyme form those chemical handshakes it's looking for. And fourth, those handshakes flip the switch and activate the enzyme's molecular scissors. The con is complete. The big takeaway here is actually pretty mind-blowing. The water inside our cells isn't just some passive background liquid. It's an active participant. It's part of the system that enforces the rules of biology. And by taking it away, you can literally convince a molecular machine to break the very rules it was built to follow. And that just leaves us with this one last fascinating thought. We just saw how something as simple as the presence or absence of water can completely rewrite the behavior of a complex machine inside our bodies. So it really makes you wonder, what other invisible forces, what other parts of our cellular environment are quietly shaping the fundamental rules of life right under our noses?